Okay, welcome and thank you for joining us today. I am Carlos Vasquez, FASE's CEO. At FASE, our focus is to understand, translate, and intervene with food allergies. And we have established a unique collaborative network of leading scientists focused on bringing us one step closer to a cure. In a short period of time, FASE has changed the landscape of food allergy research, and we're excited for the road ahead. This year, we put together an exciting lineup of speakers for our seminar series, Our Journey to a Cure. Throughout the year, we have been discussing FASE's endeavor to create a world without food allergies. Please continue to join us on our monthly seminar series, where we'll spotlight critical science on the path to developing transformative therapies. Today, we're thrilled to be joined by Dr. Noah Palm, new investigator at FASE. And Noah will be talking about bugs and drugs and how to screen gut microbes for interactions that benefits host and immune reactions. Noah is an associate professor of immunobiology at Yale University of Medicine, and his lab has been focusing on understanding how trillions of microbes that live in us and on us interact with their host, and how his team is developing new technologies to convolute this complex host microbiota interactions and how that impacts human health. Before we start, I just want to remind everyone that we'll be taking questions from the Q&A box at the end. So please submit your questions along the way, and we'll be ready to ask the questions and answer the questions at the end. So Noah, great to have you here. Really a pleasure, and really excited that you're now part of the FASE network of great investigators. Before we start, Noah, I think it's always great for our audience to get to know you a little bit better, not only about your science that you're doing, but your background, how you got to where you are, how you got involved with FASE, and why food allergies.
Yeah, great. Thanks, Carlos. So I trained as a basic immunologist here at Yale, where I'm still where I'm faculty now. But about 15 years ago, I really became fascinated with the emerging data suggesting that these microbes that live in and on us, our so-called microbiome, plays a really critical role in shaping various aspects of host immunity. And so I've really been focused on that question of understanding how those microbes influence us, really trying to understand that at a molecular level. And my lab really is now kind of a mixed microbiology and immunology lab, focused on really trying to uncover the molecular mechanisms that explain the profound impacts of our microbiome on nearly all aspects of human health and disease. So my interest in allergy, though, actually goes way back to my graduate studies. And I actually did my PhD with one of the scientific founders of FASE, Ruslan Medzitov, where we were working on trying to understand the kind of evolutionary origins of allergic immunity. So really why kind of these pathways that enable allergic responses may have evolved in the first place. I also have actually a personal interest in allergy. Because my wife actually, I don't have any food allergy. Luckily, I have an 8-year-old son, but no food allergies yet, as far as we can tell. We gave him lots of peanut butter when he was a baby, which I think hopefully worked. But my wife actually suffers from an extremely severe allergy to latex. So to the point where actually she'll have potentially anaphylactic responses to being in the same room as latex balloons. Or actually in the same room as gloves, and even will have allergic responses to food that's been prepared with latex gloves. So it's not exactly a food allergy, but I think because of that, we've really understand quite well some of the restrictions that come with these kinds of severe anaphylactic responses. So, like probably many people maybe on this call who have children with food allergies, we don't eat out unless we've vetted the restaurant beforehand and what kind of gloves they're using. And even travel can be difficult because in some countries they still use latex gloves in their airports. And so, this is one reason that I kind of have a lasting interest in personal interest in allergy as well. And so I've been extremely grateful to FASE for allowing me to join kind of on this important effort and be able to return to kind of one of my first scientific loves, the allergic response, and hopefully contribute to providing actionable insights into how the microbiome may actually contribute to development of allergic diseases, or perhaps even protect from development of allergic disease.
Thanks for sharing that, Noah. No, I think actually learning about your case in your family with food allergies has been one of the most interesting and more challenging cases that I've heard from our network of people experiencing food allergies. Just being able to just experiencing being in the same room with someone with latex and having those reactions is just crazy. No, the microbiome, we know there's a lot of advances happening in the microbiome, understanding the microbiome, and we know that the gut microbiota is a complex community of many different microbes that interact among themselves and also with the host. So there's a lot of advances. Can you start by providing some background to our audience about what is the microbiome and how complex it is? That would be great.
Yeah, of course. So some of you may kind of have heard some of these numbers before, but I think it bears repeating just to really emphasize the complexity and diversity in the microbiome, and therefore kind of the complexity of the problems that we face when trying to understand how these microbes actually influence us. And so probably many of you know at this point that we're constitutively colonized with trillions of microbes, most notably in our gastrointestinal tract, and that each of us harbor communities consisting of hundreds of species that actually encode millions of genes. So this is about 100 times more genes in an individual human's microbiome than there are in our own genome. And actually, these microbes also have important metabolic effects. So they produce tens of thousands of unique small molecule metabolites, either de novo by production of metabolites, or by processing and changing the chemicals that we eat either as food or as medical drugs. And if you look at this kind of at a population level, the complexity is even more overwhelming. So, there may be tens of thousands of species across the collective human microbiome, with each of us harboring kind of a unique microbial community. Some people refer to this as microbial fingerprint, encoding hundreds of millions of genes and potentially producing hundreds of thousands of metabolites. And we found of particular interest to immunologists, a lot of studies over the past few decades have revealed that these microbiomes really play a fundamental role in educating and shaping our immune system, both in the gut as well as in distal tissues. And so, for example, these metabolites I mentioned, some of these metabolites produced by gut microbes can actually accumulate in systemic circulation at up to millimolar levels. So this is more than almost any medical drug, so potentially allowing them to influence diverse tissues. And in terms of effects on the immune system, we also know that different microbes can push and pull the immune system in different ways. So certain microbes can induce inflammatory responses, while other microbes can actually suppress those inflammatory responses. So because of these kinds of profound effects of the microbiome on the immune system and the importance of the immune system in so many diseases, it's perhaps not surprising that changes in microbial communities have been associated with basically every disease you can imagine. Now, this is often referred to, imbalances in microbial communities are often referred to as so-called dysbiosis. And, allergy is not an exception to this rule. There also are many reports of associations between alterations in microbial communities and atopic individuals. And so in the case of kind of food allergy in particular, there are multiple ways that the microbiome can potentially affect, for example, sensitization, so development of food allergies. And I think kind of the emerging paradigm is kind of the idea that a so-called healthy microbiome, although I should say we still don't really know what is exactly how to define a healthy microbiome, other than knowing that when it goes out of whack, bad things happen. But the idea kind of is that healthy microbiomes may protect against allergic disease, so that these healthy microbiomes actually enhance the barrier functions of the gut. So they will lead to exclusion of these kinds of antigens, preventing them from getting across the barrier and potentially inducing allergic responses. They can also directly promote immune tolerance, so may induce tolerance to these foods, and probably provide signals that even if we make immune responses, that skew them away from making allergic type immune responses and to making other kinds of immune responses. And conversely, in dysbiotic states, microbes can lead to exactly the opposite kind of changes, different microbes, where you have defective epithelial barriers, sometimes referred to colloquially as leaky gut, which many of you may have read about in the popular press, which just means that there's gaps between epithelial barriers that then these antigens can leak through, leading to loss of tolerance and eventually potentially allergic sensitization.
Thank you, Noah. Especially, there's been a lot of new understanding on the microbiome, and it seems like a lot of studies, many of the studies are focused on identifying the best bacteria and kind of as an entity. But it seems one of the interesting things in your lab, it's also really looking more synergistic effects between different bacteria and how they communicate. Can you talk a bit more about that? And also, I think there's been a lot of progress, yet the question always comes, well, why don't we just use bacteria to solve food allergies? So I don't know if you can touch on those 2 points a little bit more. That would be great for our audience.
Yeah, so in terms of kind of the challenges that have made it hard just to take bugs as drugs and probiotics, for example, or next generation probiotics and use them to solve food allergies, I think there's a number of reasons that's been hard. Some of them go back to kind of the fundamental challenges in the microbiome field. So one of these challenges that's kind of a cliche at this point, but bears repeating, is that correlation does not equal causation. So these tools, technologies that have been developed that allowed us to catalog all of these microbes and their genes and their products allow us to find lots of associations between changes in microbial communities and say allergic individuals, but that doesn't mean that these changes are causal. And so this has been one of the major challenges in the field, is to know which correlations we should actually be focusing on. The other big challenge we've faced, as I talked about all these genes and all these metabolites, is that, again, these kinds of technologies that allow us to catalog genes actually aren't very useful for telling us what those microbes or genes do. And so even though we have these catalogs, we actually still don't have annotations for the vast majority of these genes or metabolites. Sometimes people refer to this problem as the dark matter of the microbiome. And the magnitude of the problem means that these kinds of traditional scientific approaches, like one-by-one approaches, where you go in and pick one gene, assign it to a graduate student, they knock it out or knock it in and see what happens, are not suitable because we have 200 million genes that we have to deal with. So even, and then kind of coming to the bugs as drugs approach, even when we actually identify microbes that we think might be helpful, there are still multiple challenges. One of them is inter-individual variation. So one bug may work in one person because they have these unique microbial fingerprints, but may actually completely fail to colonize another person. And this is the case for many probiotics, and it has been a major challenge in the field. In food allergy in particular, it's also not clear when we would need to intervene, when actually, when in time we would need to intervene. And we don't have good biomarkers to know which individuals at which time points we should be putting these bugs as drugs in. And the final kind of challenge is maybe a little bit more prosaic, but still really fundamental and important, which are the regulatory challenges. So this idea of using bugs as drugs is, for the FDA, really a new way of doing things. And so there's all these kinds of regulatory hurdles that need to be gone through. And that's meant that there have been a lot of delays in development of microbiome targeted therapeutics, some early failures actually in microbiome therapeutics. And for that reason, especially lately, actually money from venture capitalists for investing in microbiome targeted therapeutics has been drying up almost completely. And so I think this is another challenge we're going to face in the next 5 or 10 years. And maybe one reason that organizations like FASI that believe in this hypothesis and are willing to, put money behind it are going to be so important in lieu of this kind of venture capital funding being available.
So not even if you had 200 million graduate students, one for each gene, you would be able to solve that question. No, it's interesting, and that's a very good point. I have seen that trend now in the industry, given a lot of the challenges. I feel everyone felt that we have understood it, and the microbiome was so great, we can go and solve diseases, but it's been very clear that there's so many questions to remain unanswered. But there's so much potential in the microbiome. That's one of the reasons why we're so excited about your work. So extending on that, Noah, I think it seems like you have a very unique approach to tackle this problem and this field. So can you share a little bit more with our audience the approach that you're taking to solve this complex issue of the microbiome and their association, and not only characterizing, like you were saying, their genes, but their function as well, and the function of the metabolites? If you can share a little bit more about that, it would be great.
Yeah, of course. So one of the things that we've been trying to do to kind of sift through all of this noise is develop new technologies. I won't go into the details about how these work today, but new technologies to accomplish exactly what Carlos described, which is to kind of fish out individual microbes or metabolites that are interacting with the host in some specific way at a molecular level, and therefore potentially playing outsized roles in shaping susceptibility to disease. And one of the ways we do this is by developing new kind of ultra high throughput screening technologies that allow us to look at thousands of potential interactions between microbes and the host. And so over the years, we've developed systems, for example, that allow us to pick out the individual microbes from, say, the 500 species in your gut, Carlos, that are actually playing really outsized roles in shaping the immune response. I'll tell you a couple examples of that on the next few slides, in both good and bad ways. But also, we've been trying to dive deeper into trying to understand mechanisms of action. So developing new tools to screen microbial metabolomes, so metabolites produced by individual microbes, against hundreds or even thousands of host receptors that control various aspects of our physiology, as well as other kinds of functional screens that I won't have time to tell you about today. The other thing we spend a lot of time doing is establishing really foundational tools to transition from identifying these kind of candidate microbes or metabolites to actually testing causation using animal models. So we spend a lot of time actually growing bugs. This is using an approach that some people call culturomics, which is really just a fancy way of saying we try to capture individual microbes from the microbiome in monoculture so that we can use them as experimental tools. And then also this kind of tool that's gained a lot of popularity over the past decade or so, called human microbiota-associated gnotobiotic mice, which just means we take mice that we raise as germ-free, so that have no microbes of their own. They're raised in bubbles and fed sterilized food and water. And we can colonize with them with communities of microbes or individual microbes that we think may be doing interesting things. So just as one example of that was published relatively recently, we developed this technology to identify immunogenic microbes, so microbes that are inducing immune responses in humans. And so in the case of an inflammatory bowel disease patient, we were able to actually identify one really unique microbe that we thought might be doing interesting things to the immune system in the gut, and actually use one of these kind of simplified gnotobiotic models that's either colonized with kind of a mock community of microbes, a background of microbes that don't really have strong immunological effects, and then add just this one bug, kind of like a knock-in or knockout gene approach. So we're adding one bug at a time. What we see is that adding this one bug completely sensitizes these mice to development of intestinal inflammation. So we give them a chemical to induce an inflammatory bowel disease-like disease, and only these mice that have this one bug added develop this really severe inflammation. But we wondered if we can kind of if microbes can push us down this bad path, can they also pull us back into a good direction? And so what we did is we went and looked at what are the microbes in healthy humans that are actually inducing immune responses. And we isolated one of those microbes, and we now added it on top of this kind of bad bug to see if a good bug could reverse those effects. And indeed, what we saw interestingly is that just now adding this one bug on top completely neutralized the inflammatory effects of this Alobaculum species shown here. So this is a marker of inflammation that we use, so a marker of inflammatory bowel disease-like syndrome in these mice. And you can see now when we have both of these microbes together, it completely blocks disease as well as blocks actually some of the markers of immunity. This is one marker of eliciting inflammatory responses that we often use in the gut. And again, you can see that this one bug drives a really strong response, but if you add another bug on top, it actually blocks that response completely. So the final kind of example that I'll give is an example from trying to dig deeper into not just which bugs are doing interesting things, but the mechanisms by which they're mediating those effects. So again, I mentioned that we've been developing these technologies to screen for activation of potential metabolite sensors. So one family we're really interested in are the G protein coupled receptors, which some of you may be familiar with because they're the largest family of transmembrane receptors encoded in the human genome. There are over 300 receptors, actually conventional GPCRs. We've developed pooled screening technologies that allow us to screen individual metabolomes from individual microbes against all of these receptors in a single well of a 96-well plate. So I thought I'd share some kind of actually unpublished data. I've never shared this publicly before, so this is the first audience that gets to see this, that we found this really interesting bacterium that's actually enriched in infants, particularly healthy infants, that is making a chemical that's most famous for being a neurotransmitter, right? So acetylcholine. So it's actually pumping out tons of acetylcholine. And what we've found is actually that this acetylcholine production is driving, is actually facilitating the production of this specific secretory antibody called immunoglobulin A, something we've actually been working on for a long time. And we were able to show this because we were able to find in this microbe the pathway that's necessary for production of acetylcholine and actually knock out the enzyme that enables production of that acetylcholine. And what we could show is that actually the mice colonized with a knockout had significantly less expression of the heavy chain of IgA, so this particular antibody isotype, as compared to mice colonized with the wild type bacterium. So we're really attracted to the idea, because IgA is actually really important for tolerance to the microbiome, but also perhaps tolerance to food. We're very attracted to the idea that these kinds of signals coming from the microbiome in early life may actually promote tolerance to our microbial friends as well as dietary antigens. And that's a hypothesis that we're working together with you guys to interrogate right now.
Not only it's interesting the effect that you're seeing when you're putting another bacteria, but the fact that you were able to identify that a neurotransmitter that it's generated by a good bacteria can lead to protective mechanisms induced by another part of our system, the immune system, with IgAs, another antibody that our body generates. It's so exciting. I think it really goes to connecting to some of the points that we've discussed in the past. When you were looking at this problem, it's not only looking at the immune system, but looking at the microbiome now, neurotransmitters. Great to see this story and how things are evolving. Amazing findings. No, I think this has been fascinating. I know we only have a few more minutes, so I'll just, before we go to questions and answers, I know we have a few questions already in the Q&A box, but before I do that, I just want to remind people, first of all, thank you for this great talk, Noah. Everyone asks us all the time, how can they get engaged with FOSSI? You can donate, you can host events, follow us on social media, join our next webinar, and here's a QR code that you can use, easy to use to connect to us. Don't miss the next webinar we're going to have, where Dr. Ramnik Xavier will be talking about more of the transforming single cell data that his lab has been understanding to characterize the gut architecture in food allergies, and that will be in April 2nd. So with that, we can go to the questions and answers. And no, I think the first question that I have here is, hey, what's next? You're trying to tackle this big question, and it seems like it's so large and so diverse. What's next in your mind, and how to start really teasing apart this amazing question of the microbiome, and especially in food allergies?
Yeah, so I think I could go on and on about that, but I'll try to be somewhat concise. I mean, I think that what I've shown you is that we've started to make this kind of transition from correlation to causation and even to understanding some mechanisms using animal models. But these are really still at the proof of concept kind of stage. Sometimes I even say they're kind of collection of oddities stage. We're collecting examples where microbes can do interesting things to us. But we really need to really translate to human populations. We really need to understand, how much the microbiome matters relative to other environmental and genetic impacts. And of course, we want to target it only if it's a major contributing factor. In which individuals is it playing a major role? And at what times is it, as I think I mentioned also earlier, at what times is it most important? I think also another one I alluded to, or maybe my data highlights a little, the data I shared highlights a little bit, is the combinatorial challenge. So that you saw we can add one bad bug, make the mice sick, but we add another good bug and the mice are healthy again, right? So that kind of understanding how actually not just individual microbes, but combinations of microbes lead to different outcomes is another major challenge. And then the final one is that I think just in general, as you mentioned as well, people have been quick to try to rush into the clinic with crude approaches to solving the microbiome, or what I would consider crude, like transplanting whole fecal microbiomes from healthy individuals into sick individuals. That's worked for some infections like C. difficile infection, but I think it's going to be much, much more complicated for these kind of chronic diseases, and that my kind of position is that really to solve this problem, we need to get down to understanding molecular mechanisms and perhaps even moving away from targeting the microbiome itself to actually targeting the host pathways that the microbiome engages, perhaps even with more conventional therapeutics like small molecules and antibodies that we know how to develop and deliver and get approved. And so I really think that the key is understanding mechanism, and that's what's going to unlock kind of the power of the microbiome therapeutically.
Great. I think we have time for one or two more questions briefly. One of the questions that I have here is, are all these bugs aerobic bugs, which will be more in the small intestine, or do you also, or are there some that are anaerobic bugs, mainly in the large intestine, responsible for the pro-inflammatory states? And if it's anaerobic bugs, is that difficult to handle in aerobic conditions in the lab?
Yeah, so maybe I should have mentioned this. So most of the bugs we work with are anaerobes, and we grow them in specialized bubbles on specialized medias that allow us to cultivate these bugs, some of which will die very quickly upon exposure to oxygen. Probably a lot of the bugs that have interesting effects on the immune system are kind of in the middle. So they're not obligate anaerobes, but they're facultative anaerobes. So they can grow in both environments, which is probably what allows them sometimes to grow close to our epithelium. But the kind of protocols we use, we act to cultivate bugs, encompass all oxygen environments, so anaerobic, microaerophilic, and aerobic. And so we're really trying to cast a wide net.
Awesome. We have a few more questions, but I know we're out of time, Noah, so we'll try to address this via email if we can. But Noah, I wanted to thank you. Great talk, really fascinating area, which there's so many questions, but I'm really optimistic that with the research you're doing and others in this field, hopefully some of those questions will lead to therapies in the near future.
Thanks again, Carlos.
Thank you.